welcome to the first webinar on Invivocue's capabilities. This will be the first webinar in a series of webinars that will talk about humanized mice, technologies related to humanized mice, as well as services related to humanized mice. To have a brief introduction of Invivocue, Invivocue is actually a subsidiary company of Invivocue. We are based in Singapore, and our goal is to actually accelerate the decision for healthcare and the pharmaceutical industry using our 3D cell culture models, technologies, as well as humanized mice technologies. So our mission is to actually develop predictive in vitro and in vivo translational models to enable the healthcare and pharmaceutical industry to create better, faster, more affordable disease treatments for a better society. From 2019, which is when we started, we've had a lot of projects with pharmaceutical companies as well as international clients around the world who are interested in our humanized mice technology. We've completed more than 50 projects, used more than 2,000 mice, and we've worked with 2 out of the top 10 pharmaceutical companies in the world. We work closely with a lot of the institutes in Singapore to understand whether they would like to use our technologies to develop some products or to even understand certain aspects of biology. We've also had several long-term collaborations with some of them, spanning more than 2 years. So our animal facility, which is located in Singapore, is ALAC accredited, and we adhere to all the ethical guidelines associated with this ALAC accreditation. So we fit into the drug discovery pipeline. For people who are not familiar, the drug discovery pipeline usually starts with a target and a target identification. So you would like to see whether this biomarker that you are looking at is expressed and is, of course, a valid target for your drug therapy or other modalities, for example. From there, you progress on to a hit. So you try to develop a drug or therapy that actually works on this particular target. And when you get to the lead optimization, this is where you try to modify parts of this particular therapy to suit your need in this case. Where we fit in is during this lead optimization phase, as well as in vivo testing phase, where we cover stuff like absorption, distribution, metabolism, excretion, ADME for short, target identification and validation in the model itself, rather than just a Petri dish, and in vitro. And we handle the in vivo testing of these particular compounds as well. So pharmacokinetics, toxicology, as well as efficacy are some of the services that we offer using our humanized mice or other models. So in recent times, there has been a push towards developing better models and developing more translational models. Our goal by developing humanized mice is to actually reduce the number of mice that you need to observe an effect. That's one of the key goals that we have in humanized mice development. By developing a humanized mouse, you would be able to have parts of, let's say, a human system expressed in this particular mouse that allows you to study the effects of whatever drug target or therapy you might be interested in, working inside a human system without actually testing it on a human. So this allows you to bring to the market a more translational and more beneficial approach to the drug discovery or the drug development pipeline. The FDA has indicated that there is a need to replace, reduce, and refine animal testing according to the 3 Rs. And by using humanized mice, you would also reduce the number of animals that you would need for testing, for example. To give a brief overview of humanized mice, there are previously transgenic mice as well as non-humanized mice. A lot of transgenic mice generally lack a lot of the features and expressions of certain proteins or certain receptors in this particular model. So they lack the human aspect of this. Some of them incorporate the human aspect to a certain degree, but for a lot of the other functions of that particular cell type, for example, they don't have the human receptors, and they don't have the human proteins associated with those particular cells. Where our technology comes in is when we inject and we are able to reconstitute the liver and the immune system of the mice to be that of a human. So they will start to express human immune system cells, such as T cells, B cells, and of course, they can also express human liver cells as well. This allows you to understand a lot more of the mechanisms of human biological responses that might be human specific, such as genetic or immune factors, microenvironments, for example, that are specifically related to a particular disease or particular disorder that has that genetic component that's only replicable in a human system. A lot of infections that people intend to study or are studying, such as HPV or HIV, are human specific and they only target human cells. So you would need a human system to try and understand how the immune system interacts with this particular virus, for example. And of course, it cannot be done in humans because that would be unethical. So another aspect of this is that the heterogeneity that comes with the human samples are also captured in this mouse model. Because every human is different, this platform allows the development of personalized medicine to a specific population or subpopulation of humans that have this particular mutation or biomarker, allowing you to have a more personalized medicine development. You can stratify your drug targets in terms of who might be receptive to this particular therapy. And this allows you to develop a more accurate treatment response, pharmacology, and you can observe drug side effects that you might not observe in a mouse system, for example. These are an overview of our capabilities. On the in vitro side, from Invivocue, we have patient-derived organoids, in vitro studies using immune cells, cardiovascular, skin, and hepatocyte cell cultures, as well as 3D cell cultures such as 3D cancer organoids, 3D airway models, as well as 3D hepatocyte Kupffer cells. On Invivocue side, we have the humanized mice platform, which contains humanized immune system mice, or HI mice for short, humanized liver mice, or HEP mice for short, and the dual humanized mice, which combine both the immune system and the liver of the human into a mouse. We call this the dual humanized mice. What we can use these mice for is actually cancer, graft versus host disease, ARDS, inflammatory bowel disease, non-alcoholic steatohepatitis, or NASH, systemic lupus erythematosus, or SLE for short, and immunotoxicity. These are some of the key areas that can be that leverages human mice. To break down our humanized mice models into more detail, I'll start with the high mice. The high mice are built on a immunodeficient mouse strain, the NODSKID IL2RG mouse model. And from there, we reconstitute the human immune system with a CD34 positive hematopoietic stem cells. These are derived from cord blood. So a lot of the cells will start to reconstitute into this mouse system um, and develop into T cells, B cells, myeloids, and NK cells, along with other cell types. In this case, the liver of the mouse is still non-human, and only the immune system is humanized. There is no GVHD present in this mouse model, and they live to a long lifespan that allows you to study a lot of the immune effects that you would not be able to study in a GVHD model, for example. So the next one would be the HEP mouse, which is a mouse model repopulated with human hepatocytes. This is also built on an immunodeficient mouse model strain, which is the NODSKID mouse model, together with a FAH knockout background. So this allows the reconstitution of the liver as they slowly get cleared from the body with human liver cells. So in this case, the human immune system is not present in the mouse, but the liver is more than 70% human. This allows you to study effects related to, let's say, pharmacology of a drug, where metabolism, by the human liver would be different than that of a mouse. And lastly, we'd have the dual humanized mouse. This combines both the human immune system as well as the human hepatocytes into the mouse model, which allows you to study the interaction between the liver as well as the immune system, which for certain cases such as HBV or HIV, these would be the ideal model to take a look into the human immune system interaction with the liver. For the humanized immune system mice, we are able to modulate the immune system to a further extent as well. Because reconstitution may not be sufficient for a certain experiment, for example, we would be able to modulate the immune system to express more or less of a certain type of immune cell. In this case, it would be myeloid improved cells. So these would express more myeloid cells, as well as NK cell improved mice. This allows you to study more of the NK cell population or the myeloid population. In the myeloid population, you see more T cells, B cells, monocytes, as well as a bit more NK cells. But in the NK cell improved, the main focus is on the NK cell, and this allows you to express the NK cell much higher than would be seen in the myeloid improved cells, for example. So some of the applications, as I mentioned previously, that we cover are oncology and so on. But I'd like to go through a bit more of the in-depth applications of high mice that we've had interest for, as well as what we are currently working towards. So as briefly mentioned just now, we have a focus on oncology, ARDS, SLE, GVHD, IBD, NASH, CRS, and immunotoxicity. For the sake of time, today I'll only be covering a few of these disease areas. And at a later date in a subsequent webinar, we will be covering more details related to these diseases. I'll start with the oncology aspect. So combining our technology from Invivocue, which allows us to take human samples, human cancer samples, allow them to expand and grow into patient-derived organoids or PDOs. We are then able to put those PDOs back into the humanized mouse or a different mouse strain, and we can observe the development of this particular tumor. This particular tumor will be specific to that particular patient's cancer. This allows you to look at treatment effects or if there's any metastasis present in this particular patient tumor sample, which allows you to test and screen for several types of different drugs, or if you're trying to develop a therapy for this particular type of cancer, this would be of interest. So we have a wide variety of access to PDOs, such as, let's say, breast cancer, ovarian cancer, lung cancer, colon cancer, with different biomarkers associated with each of these particular cancer subtypes. So the PDOs that we obtain from patients and then we proceed to grow them in the mice, they do express and retain the phenotypic characteristics of the original tumors. This allows you to study, even if unfortunately the patient has passed away for whatever reason, to study this particular cell line such that you can prevent this particular cancer type or subtype from killing another patient, for example, if you can find a treatment for this. So we have a wide range of PDOs available in our biobank for commercial use as well. Some of the biomarkers, as you can see on the screen, are captured here. And this profile allows you to develop a targeted therapy, or if you have a drug screening library that you would like to test on specific biomarkers, this would also be a possibility. We characterize the growth of some of the PDOs inside the mice models to observe and understand how long it takes for the mice to develop, let's say, a particular tumor to a certain volume. This gives you an indication of how aggressive the tumor would be and how likely the tumor is to metastasize, for example, or even after the treatment, relapse. In addition to the patient-derived organoids, we carry out experiments related to cell line-derived xenografts. So these would be your typical cancer cells, I mean, such as NCLN87, gastric carcinoma, pancreatic adenocarcinomas, as well as other solid or liquid tumor cell lines like NALM6, MDAMB231, HCT116, HT29. So we're not limited by these number of cancers or cell lines. When we have worked with a lot of different cell lines, and we are very experienced with understanding what it takes to develop the right therapeutic if you're testing on these particular cell lines in the mice. One more key area that we focus on is actually autoimmune disease. We focus quite heavily on systemic lupus erythematosus, or SLE, which is an autoimmune disorder characterized by nuclear antigens as well as inflammation in several different parts of the body. So we've developed a humanized mouse SLE model, which is specific to the human immune system. So a lot of the phenotypes that you see would be human specific, as opposed to other mouse models that may not have the human component. So you would be able to test if this particular SLE therapy would be able to address a lot of the phenotypes that you see in humans. So some of the phenotypes that you see are the increase of the human autoantibodies, lupus nephritis, lymphopenia, activation of the B and T cells, and of course, the inflammatory cytokines circulating within the body. We use a pristine mouse model in the humanized mice, and we observe a lot of these features with very, very strong and robust phenotypes that are consistent across several experiments and several studies. And lastly, I'd like to talk about some of the partner services that we offer in Invivocue. So we offer, through our partners, testing in GLP, non-GLP toxicity, behavioral assessment work in the areas of neurodegeneration and neuroscience, as well as intravital microscopy that allows you to observe what changes in the mice without needing to sacrifice the mice. This allows you to observe the mice on a cellular level, depending on which organ you might be interested in. And you can observe aspects such as drug bioavailability, as well as microenvironment in the tumor, for example, and the immune cells that migrate towards this tumor microenvironment. So that brings me to the end of this webinar, and I thank you very much for your attention. So if you would like to reach out to us, please do contact us at the following address over here. Thank you very much for the time, and I hope you have a nice day.